everyone. Thank you for inviting me back and for allowing me this first slot. The SNP markers and the DNA fingerprinting is not everybody's cup of tea, but I hope I'll engage you and give you some more progress of what I presented on the last time that I was here. So the title is The Genetic Fingerprinting of Cannabis Strains for Enabling Future Validation. So as I was introduced, I'm Inga Gazendam, and I'm from the Agricultural Research Council, the institute that's just north of Pretoria, the Vegetable Industrial Medicinal Plant Institute, close to Rurapla Dam. So just a very basic introduction slide for the involvement that ARC is. Anything that you can mention in cannabis we are interested in doing research of. So there's various aspects that I'm not directly involved with, but we have a huge DALRAT project where almost any discipline is being researched. So we are involved in various aspects, the fiber and the oil or hemp cultivars, but also medicinal and recreational accessions. So our focus is on breeding, evaluation trials, propagation, cultivation, disease diagnostics, and also quality assurance. So to start off with a basic introduction of what genetic fingerprinting entails, firstly, a genetic marker is a specific DNA sequence that you have a known location of on a chromosome, and the different genetic variations that we find between different individuals can be picked up by measuring this DNA marker. So for fingerprinting, we use these markers to characterize the genetic differences between lines and cultivars. So why is it useful for cannabis? So what we aim to offer is to validate the authenticity of strains. The trueness of type is another way of saying that. So for specifically clonally multiplied plants, they will have identical genomes. So the DNA fingerprinting will have an identical profile. But also if you have your strain that you want to protect for cultivar protection, you need to show that it is the same as what you've measured before. And as I was introduced, we are building up a database of strain genotypes to help with capturing that data. So the advantages of doing DNA fingerprinting is normally you would characterize them morphologically, but there's a disadvantage to that is because the different strains sometimes look very similar, and it's very labor intensive to grow and morphologically characterize strains in the field, and they can differ from season or from environmental conditions. But the advantage of DNA is it always stays the same. So you can distinguish between these similar accessions by looking at the DNA. It's not influenced by the environment, so the stage of the plant, the growth stage, also doesn't influence when you genotype them. You can use it from a very small in vitro plant or a seedling, or even we've done it from dried leaves as well. So this is the interesting part to me, but might pass by on my audience, but I want to show you how neat the technique is. So the technique is based on simple sequence repeats. They are distributed through any plant genome, and they are short fragments that are duplicated in variable numbers. So if you PCR them with flanking primers, you will find fragments of different size lengths. That's just a diagram to show how the number of repeats can differ between this marker. And because they are short repeats, they change during replication. So over evolution, they will become variable numbers, and that's what we use in the fingerprinting profile. So we use a published set of 13 SSR markers that's commonly used for forensic cannabis profiling, and it works very well in our lab as well. So the steps that we would perform in the lab is when we receive the leaf sample, you extract the DNA and do the polymerase chain reaction in a PCR machine. We utilize a service that analyzes the fragment for us on a genetic analyzer. The output is peaks that's nicely different, colored according to the fluorescent dyes, and we score those sizes of peaks and create the table of allele sizes. So the neat trick of this method is also that we can analyze all 13 of these markers in one tube. So they are fluorescently labeled with 4 different dyes, and we choose them specifically so that the fragments occur in non-overlapping regions. So you can throw them all together in one tube, and then when the fragment analyzer analyzes the peaks, they occur in different spots, so you can assign them to which marker they are. So this is just how the 13-plex panel is distributed with the 4 colors of dyes. And multiplexing is the word that we use to say that we analyze them all in one tube, which enables cost reduction but also effort and time. So this is the neat machine that the capillary electrophoresis step is run on. So those differently labeled fragments are separated in size in a column, and when they pass at the photodetector at the end of the instrument, it gets detected and the size with an internal standard gets determined. So the different 4 dyes then end up in the peak. As I said, the different fragment sizes are those peaks. So this is just a screenshot of what the database in Excel would look like. The numbers are just the allele sizes, like the size in a little number, colored with Excel conditional formatting. So this is how we can keep our database and to see if lines are identical or not. So there's no, it's very reproducible. We can do replicates of clones and find them 100% identical. So this is just a screenshot of a few of the genotypes that we already have in the database. So our database contains 550 accessions so far, but it's a combination of everything that ARC is working on and also samples from clients. There's medicinal cannabis in there, collected land races. We've imported some Chinese hemp cultivars and then also hemp crosses. And there's a huge amount of diversity in them. And this circle diagram that you see there is a dendrogram just in a circular form to show that there's it's a big database, it's difficult to visualize, but we haven't found any accession to be identical to any other one so far. And part of the reason for that is most of the cannabis that we work on is seed multiplied, so of course there's cross-pollination, so there's lots of genetic diversity. So this is a diagram in a linear format, the same kind of dendrogram, but just a small snapshot of some of the accessions. So as I said, the genetic diversity is detected in any accession we've tested so far. So that means the panel of 13 markers is able to detect lots of variation. So we won't easily run into the problem of not being able to distinguish between strains. And the seed multiplication, they cause the internal intragenetic variation within strain genetic variation. But this is an example, for example, where you have clonally propagated plants, where if on the tree of the branch there's a straight line, vertical line, it means those clones were identical, and that is what you expect. If you've multiplied them in vitro, you wouldn't want to see anything else than that. And that's useful for then if you look at them down the line a few generations later, that you expect the genotype to stay the same. So this is just a way to visualize genetic variation, showing you that the variation is actually in clouds. There's no pure strains unless we're talking about the in vitro clonally populated ones. And then I added this last slide in a little bit. I talked about it before. It was preliminary at that stage. It's still the same 2 markers that I find is very closely correlated with genotyping between female and male plants. And that I understand is useful for the industry if they want to find out if a plant is male or female. So in the combination of the 2 markers, which I just call marker 8 and 9, I with a very high confidence can distinguish between female and male. So I've tested it on plants that were phenotypically characterized as female or male. And for example, the green dots there are all expected to be male, so they fall in that cluster. It's a scatter plot of SNP markers. So the male ones are normally heterozygous, and then the females are the homozygous of the one allele. So the unknowns we could successfully then score according to in what cluster they fall. And this is another experiment with the same marker to show that the green dots again is the males that we expect there, and the red ones and the yellow ones are the unknowns ones that we could assign a genotype to. So in conclusion, our molecular tools are handy tools to detect genetic differences and removes the disadvantage that you have with morphological characterization, since strains can look very similar and also will be very varied. And our database is growing. The ISSR alleles database is growing with every accession that I do. And we can provide these services to the industry. So you can contact me if you have samples that you want to genotype to check their authenticity later on in your process, and for legal protection if you may need that. So my contact details are there if you need to email me to ask for what we can assist you with. And then my colleague that's also at ARC, she has a project, she's Dr. Elsie Kruijwagen. She's involved with disease diagnostics, and she's extending an invitation for any people with disease samples in glasshouse growth conditions. They actually want samples with diseases to detect, for example, powdery mildew strains. So you can contact her as well if you have glasshouse grown samples with diseases, and she can test them for you at this stage free of cost, because they do have a project that they just need disease samples for. Thank you very much.
Thank you very much, Inga. Any questions? All good? Okay.
Hi, my name's Pierre van der Hoven from a company called Baobab. Have you started looking at the landrace strains from Mpondo land, for example? And if so, how far are we down the road of actually knowing what a landrace strain is?
My only answer I can give is yes, I've done land races. All I can see is they are very variable, so I can't identify accessions in groups, in clusters yet. Even the cultivars, hemp cultivars, is very varied. So the SNP markers or the SSR markers are actually too good at detecting variation. So I think we must look at more uniform cultivated strains before we'll be able to cluster them. Land races, I'm genotyping whatever ARC is collecting, so I can't now for a fact say where they were collected, so I can't answer where those ones came from. But, I mean, the database is growing.
So the follow-up to that is to actually develop that. What do you need? Do you need seeds? Do you need plants? Do you need flour? Do you need volumes? How do we get to the end game of knowing what our land races are? Because currently the biopirates are running around, they're stealing our genetics.
True.
And we need to protect those. So I'm trying to establish where's the government and what's the approach and how far are we to actually protecting those land race strains?
My opinion is we're not really far, but we do appreciate that that is the problem. Material-wise, what I would say, any material, we've not done it from seeds yet, but I'm sure it's possible, but any small leaf sample, but because it's a population, whatever you collect now may not be the same population that exists in the next season. So that's a difficult part of working with seed-propagated crop, and if you sample from somebody that don't culture it in a pure field and with barriers of cross-pollination. So, I mean, the fingerprinting will be great for clonally propagated plants, but if more information is first needed, the more samples the better. Then maybe the clusters or the landrace clouds can converge, and then you can maybe identify a pattern where a landrace will fit into a certain part of the whole database.
Thank you. Any other questions? Guys, free testing? Just mentioning it. Thank you very much, Inga. Much appreciated.